you, Roger. So I always feel very inspired when I hear Roger talking. He's setting out his kind of grand vision for reversing the course of Parkinson's disease. And actually, what I'm going to do is slightly complementary and more kind of focus down on the tiny little details of what actually happens in the cells. And I really kind of approach this from the point of view of cell biology. So the question that we're asking is, what goes wrong in these dopaminergic neurons, and what kind of models can we use to interrogate this issue? So, in order to do that, we need a little bit of basic biology. So I'm going to tell you a little bit about what cells are first. So it's kind of GCSE type stuff, I guess. And then I'll talk a little bit about why neuronal cells are so special. So what makes them unique? What makes them specifically susceptible to the disease process? I'll then talk a little bit about what actually goes wrong in cells in Parkinson's disease. I'll refer to that as PD from now in the presentation. And actually this is kind of more what we actually know about it so far, I guess a review of what the current ideas and concepts are in how cells start to show the pathology of PD. And then I'll discuss briefly what I found during my PhD and in the lab of Harvey McMahon, the LMB, before touching briefly on what I hope to kind of, what I hope this sets off in the future as well. So, first questions first, what is a cell? So a cell really is one of the fundamental units of life, all life, human life, animal and plant life. And actually we all start from this fusion between a sperm and an egg cell. Essentially that is the two cells, those are the two cells that basically give rise to all the thousands, millions of cells in our body. And so these two cells fuse, you then get differentiation into the various tissues, the various organs, such as skin, hair, eyes, nervous tissue. And so you might have guessed from this that we actually have quite a lot of cells. And does anyone, I'm just going to open this out to the floor, does anyone have any idea how many cells one human being is composed of? Guess.
3 billion.
3 billion, right? Anyone else? Any takers? Just shout it out. Trillions. All right, good. So it's supposed to be
I thought it was real.
I didn't count. It's from Google. so it's supposed to be 40 trillion cells in each individual. So it kind of logically follows from that these cells are quite small. And so I'm going to try to show how small they are here by drawing my little scales. At the top we've basically got a ruler, and this measures, so 1 millimeter and then decreasing size, so every gradation here is a division by 10. So that's a tenth of a millimeter, which is 100 micrometers, a tenth of that, which is 10 micrometers, and then this is a thousandth of a millimeter, which is 1 micrometer, and then finally 1/10,000th of a millimeter here, which is 100 nanometers. Okay? And sadly for us cell biologists, what's visible to the eye goes from about 1 millimeter to about 1/10th of a millimeter. The cells generally are smaller than that. So they inhabit this range from 1/10th of a millimeter all the way down to 100th of a millimeter. The cell's made up of various components, and all the components in the cell are an order of magnitude smaller, and these are the organelles that make up the cell. Most of these we can actually follow by using a microscope. At the bottom end of this range you have very small particles such as viruses, and they are 10,000 times smaller than a millimeter, in the range of 100 nanometers, and for that you need to use different techniques such as electron microscopy. So, I think a bit's fallen off the bottom there, you can read the bottom bit there, but if we zoom in on the cell here, effectively what I'm trying to show is what a cell looks like, a diagrammatic representation. And the way I like to think about the cell is like, as if the cell were a city. That's probably the best analogy to use here. Okay? So every city has a wall around it, or a boundary, a defensive perimeter, let's say. And that is, in a cell, the cell membrane. Okay? The cell membrane encloses all the constituents of the cell. And here we have the city centre, or the town centre, which is the nucleus. The nucleus contains all the information required for the cell and all the blueprints to manufacture components of the cell that sit in the cytoplasm. So around the nucleus you have all the constituents of the cell, and I think of these constituents or organelles like factories that subserve different functions. Okay? So, for example, you've got the lysosomes here, and these are the waste disposal bins of the cell. You've got the endoplasmic reticulum here, and the Golgi, which are manufacturing plants. You've got the mitochondria, which are the power plants of the cell. And in addition, how do you get from place to place within the cell? You take roads. So that's the cytoskeleton here, right? You can see here these tracks. And what happens is different components of the cell are trafficked from one part of the cell to the other along these roads. Sometimes they may be enclosed in vesicles. So vesicles are small membrane-bound organelles that traffic along these tracks. So you can think of the vesicles like lorries that go from place to place containing cargoes. All right? But what makes a city? It's not just the architecture, it's people, right? So what are the people in this cellular city? And the way I like to think of that is the people are proteins, okay? So the proteins are manufactured here in the nucleus, and then they go out to various points in the cell to subserve different functions. So the proteins are the things that we think of as having agency. So the proteins shape and regulate the cellular environment. So they are very, very important. And actually, it's one of the fundamental concepts that we have in disease, right? So we think of actually proteins going wrong in disease, protein dysfunction in disease. And what this actually means, and we speak of this in terms of genetic defects. So the genes or the blueprints that are kept within the nucleus are defective. Therefore, defective proteins are made. These defective proteins cannot serve their normal function. They therefore go wrong, and the cell exhibits signs of disease. So just to give you a slightly better idea of what goes on inside cells on a kind of subcellular level, so this is kind of really zooming in, trying to find out what goes on inside the cell. I've got a video here of some work done in our lab a while back now, and what this shows is we've highlighted a protein, we put a dye on the protein, and you'll see it in grey. The protein is called endophyllin, and what you'll see is the protein actually works on stimulation with this ligand called dopamine to bring cargoes in from the periphery of the cell into the centre. Okay? So the protein is in grey is endophyllin, and you can see here all these little dots, endophyllin dots, that are basically trafficking in towards the centre, and we'll see a train appearing in the top right hand corner here shortly. And wait for it a bit. It's coming, I promise. There we go. So you can see these tubules budding inward from the cell periphery, and these little flicks that go in towards the centre of the cell. So this is a protein actively shaping the environment of the cell, taking part in important processes that help the cell survive. Okay? So proteins shape the cellular environment. Different types of cells have varying functions. Okay? So you look at those the blobby kind of round cell I was drawing earlier is an example of a cervical cell, a cervical epithelial cell. So this observes its function very well in that it's round and flat, and it's able to line the cervix of the uterus, whereas different types of cells may have different functions based on their shape. For example, a neuron. Okay, so a neuron is quite long and streaming. Why is this? And this is because its primary function is to transfer information from point to point. Right? So let's say from your head to your arm. Okay, so it's quite long. So the key point here is that the cellular shape is adapted to function. Why are neurons so special? So I'm going to show you a video here, and this is well, I'm first going to show you a non-neuronal cell. So these are epithelial, like the cervical epithelial cells I was talking about. What we've done is we've basically taken a dish of these cells and we've scraped or gouged out a big hole in the middle. Alright? Okay? And then what we're going to show, you'll see that hole in yellow. What you're going to see here is how quickly the cells surrounding it jump in and multiply to fill the void. Okay? So that's within a few hours, it's all covered up. Sadly for us, unlike these epithelial cells, mature neurons do not divide. So if we gouge out a neuron, its neighbours cannot jump in to replace it. Okay? We're born with a certain number of neurons. Roughly speaking, we're born with a certain number of neurons, and this is basically all we have. So they're very precious. We need to look after them. So, why else are they so special? So you can see here, this is an image I took of a neuron during my PhD, and you can see that it's very special shape, or better defined here. So in the middle you basically have the cell body, and you see these long arms extending outwards, the neurites as we call them. Now, obviously to communicate between different points in the body, let's say from your head to your arm, these neurons need to be long. So while a cell, let's say in your cervical epithelium, can be tiny, a few microns across, these can be up to 1 meter long, right? So that basically gives that particular cell a huge burden in terms of packaging and transporting materials to the end, bringing it back from the peripheries in towards the center. As such, there's a very high energy demand on these cells. And the second major function of neurons is to communicate with other neurons, and it does this via synapses. So each of these dots here, each of these hundreds and thousands of dots marks a synapse, and a synapse is a point of chemical communication between two neurons. What I have here is an electron microscopy of just one of these dots blown up here on the right to show what it actually contains. And what you can see there, the most notable feature, apart from these large mitochondria at the top here, are these small rounded synaptic vesicles that fill the synapse. And these synaptic vesicles all contain chemical neurotransmitters. And the chemical neurotransmitters are released into this synaptic cleft as marked here by the arrows, and they then hit this neuron at the bottom, causing a signaling cascade in the downstream neuron. And this is how neurons communicate, right? This synaptic vesicle cycling is again a highly energy dependent process, and it takes up a lot of energy. You can see this here. I've just shown a neuron which I plated out. So this is a rat neuron, and I've put on a dye here which shows when the neurotransmitters are released by individual synapses. So each of these dots is an individual synapse, and as you can see, this is one neuron, and each neuron has hundreds of synapses. And what we're doing here is we're electrically stimulating every time. You can see this writing in the top right. There's an electrical pulse that's delivered, and what happens is you get a release of neurotransmitter. And you can imagine that if this were to happen for the lifetime of the neuron, this is a highly energy dependent process. Okay? So the other thing, the final thing here, as to why neurons are special, is that this is a particularly human problem as well. I mean, just to give you an idea, we have almost 3 times as many neurons as our nearest relatives, a gorilla and a chimpanzee. So this is just a number of neurons expressed as a factor of 1 billion. And if you start off with, let's say, a marmoset, which is a fairly small primitive monkey, more like a rat actually, it has kind of 600 million neurons. A rhesus monkey, which is slightly bigger, contains 6 billion neurons. And as I mentioned, a gorilla and a chimpanzee have in the region of 20 or 30 billion neurons. A human has 86 billion. Right? That's a load of neurons. And then if we consider that each of these neurons has, let's say, 100 to 1,000 synapses, that's over 100 trillion synapses per brain. So this is basically what we have to protect. So these neurons are they're prima donnas, right? I mean, they're quite long-lived. They're quite high in their energy demands. They have complex connectivity. So therefore, the neurons are particularly vulnerable to insults which cause disease. I can put this another way. It's not really surprising, is it, that a neuron that's survived your whole life and is susceptible to the processes that age us. So, what goes wrong in patients' neurons with PD? So, Roger's mentioned something like this before. If we take a slice through the brain, as eye level, there's a structure that sits deep within the brain, the midbrain, responsible for movement. We take it off to one side here, and you look at a controlled subject, and you can see here that the substantia nigra, which contains the dopaminergic movement neuron, is actually coloured in darkly there. And just below that, you can see an example from a patient in which they've lost these neurons, they've lost this pigmentation. Okay? If you zoom in on this particular area, what you begin to see in patients' neurons with Parkinson's disease are these characteristic halo-like brown structures surrounding these pale pink areas, and these are known as Lewy bodies. Now, these have been described in Parkinson's disease for just over 100 years, okay? But until recently, we haven't really known what the machinery around these Lewy bodies involves or consists of. So Lewy bodies are a feature of PD. However, recently, so when I say recently, I mean in the 1990s, this was work done in Cambridge. So a group here found that the protein alpha-synuclein was a constituent of Lewy bodies. And so you can see alpha-synuclein is a protein that clumps abnormally in patients with Parkinson's disease, in the neurites, in the long nerve processes, as well as the Lewy bodies in the cell body here. Okay? So the protein alpha-synuclein aggregates in Lewy bodies and neurons in Parkinson's disease. That much is clear. And they further found that the spread of alpha-synuclein pathology happens or evolves temporally in a very characteristic fashion. So the idea here is that you basically have early disease, so not so pre-motor symptoms, in which the synuclein pathology or the Lewy bodies sit in the bottom part of the brainstem. You then go to symptomatic disease with motor symptoms, and you have the spread of the alpha-synuclein into the middle part of the brain. You finally have the movement disorder together with the cognitive disturbances, and that signifies the spread of synuclein to the cortex. So, referring back to my analogy of the city earlier, right, if the cell is the city, okay, I'd rather like to think of the Lewy body as the scene of a crime. Okay? So we found alpha-synuclein at the scene of a crime. Alright? But the question arises from this, what's it doing there? Okay? So is it a witness? Is it trying to help? What's its role there? Yeah? Or is it a criminal? Does it actually help to kill the cell? So in order to answer this, we looked a little bit further afield and asked, who does alpha-synuclein hang out with? Who are the players? Who are the protein players in the story of Parkinson's disease that can give us clues? And we looked at genetic forms of Parkinson's disease. So these are not typical Parkinson's disease cases, but there were lots of genes that have come out recently that cause Parkinson's disease. Alpha-synuclein is unsurprisingly one of them, but there's a long list of others. And by and large, there's a fairly strong link with a lot of these proteins to the powerhouse of the cell, mitochondria, which I'll come to in a minute. In addition, some of these environmental susceptibility factors or toxins that we were talking about also link directly into mitochondria. So, mitochondrial pathways are heavily implicated in Parkinson's disease, and indeed in cells in Parkinson's disease. So what are mitochondria? I like to think of mitochondria as the powerhouse of the cell. Mitochondria basically provide ATP. ATP is very important for almost all active cellular processes that take place. And all the ATP that we have is made within the mitochondria, which is here, this sausage-shaped structure. It's a double membrane around it, followed by invaginations of this membrane, and all along these invaginations you get the mitochondrial complexes which produce ATP and consume oxygen, hence the reason we breathe. And here's a picture of mitochondria in a cell. So they form this beautiful elongated sausage-like network that's all interconnected. And what I wanted to do is I wanted to give you an idea as to how dynamic these mitochondria are. So this is a kind of nice still picture. But these mitochondria here are actually very motile, and they undergo cycles of fission and fusion. Okay? So in green, what we have here is we have the tracks along which mitochondria move, the roads of the cell, and in red we have the mitochondria themselves. So you can see here extension and retraction of various processes of various mitochondria. You can see that the mitochondria is going to break off shortly on this bit there. There we go, plop. And then we can see this retracts back. You can see this shoots in there, gives it a kiss, comes away. There we go. So it's mitochondria. They're highly motile. They undergo cycles of fission and fusion. And actually, really, what we're trying to say about mitochondria is that they reflect quite well the energy state of the cell. Okay. So if cells are stressed, mitochondria tend to fragment or break up, and this is in preparation for the mitochondria being shoved into the cellular garbage bin, right? The waste disposal system. Mitochondria stressed, oh no, get rid of it, break it up, okay? So we throw these Parkinsonian toxins onto the cell, and boom. It's like the cell's going to, the mitochondria have exploded, right? You get these kind of fragments of mitochondria up at the top. You can see it's going to break into short, rounder structures and little fragments. So this is the cell trying to get rid of mitochondria. And this is a process that we call mitophagy. So literally mitos are from mitochondria, and phagy from eat. So the cell is trying to eat its own mitochondria. And this is in many ways potentiated by Parkinsonian toxins. So a lot of the proteins that we already know that cause PD, or cause genetic forms of PD, play an active role in the process of mitophagy. Taking, for example, one of them, so Parkin. If you look at Parkin, so you can see here some cells expressing Parkin, and Parkin is in green, okay? And you see the mitochondria in red, right? And I've been told that basically this one is the best one to follow, right? Okay, so what we're going to do is we're going to throw some toxins onto these cells that mimic Parkinson's disease, right? And we're going to see Parkin, the green, focus or concentrate around mitochondria, all right? So there we go, follow this. You can see the green wrapping itself around mitochondria. Yeah, I'm not imagining that, am I? So you can see there that as a protective mechanism, so this is kind of the Parkin is basically trying to get there, get to the mitochondria. Mitochondria is in trouble. It's saying, right, let's get rid of this stuff. Let's help shove this into the cellular waste disposal system, right? So actually, what we think happens with this, right, is that Parkin normally serves this function. In disease, abnormal Parkin doesn't fulfill this role. So you therefore get a build-up of abnormal mitochondria. Okay? That's the idea here, right? That mitophagy is abnormal in Parkin mutation cells. So, this begs the question, what have we found? So what we found is actually alpha-synuclein plays a very similar role to Parkin on a slightly separate pathway. Okay? So we found that when we stress cells, we can get alpha-synuclein to fly to mitochondria. These are very specific conditions of cell stress. You can see here in green we have alpha-synuclein, and the cells are stressed, and in red we've got the mitochondria. So it's a stressed mitochondria, and synuclein is forming around little dots around these mitochondria. So it seems to be kind of nibbling away at the mitochondria, trying to basically take little bits off and shove it into this waste disposal system. So if you think about Parkin, right? Parkin basically grabs the mitochondria and just shoves it into the skip, right? Okay. Synuclein is like a little kind of dustpan and brush, right? Just kind of wandering around, just taking off little bits of abnormal mitochondria, just kind of just nibbling away at it, right? And so to get a better idea of this, we wanted to image this live as well. So we want to see this in motion. Okay. So this is a small, very short video. This is a year of work to label this particular protein. Okay, so this little dot is basically alpha-synuclein, okay? And what you're going to see when I play this is the mitochondria, and you can't see any red there at the moment. There's not any red inside the dot at the moment. What you're going to see is it kind of kisses the mitochondria, nibbles it a little bit, and then at the end comes off holding a little piece of the mitochondria, okay, like a little trophy, all right? So here we go. All right, so there we go. It's running around. Oh, bump. Oh, yeah. Oh, yeah. Maybe. Oh, yeah, a little bit more yellow. Hey, so you can see there, right? So you've got some yellow. We've got some yellow there, which signifies that the red and green have come together, and we've sucked a little bit of mitochondria out of there. All right. So what we think is that alpha-synuclein is going to mitochondria that are stressed and nibbling away at the mitochondria, trying to basically rescue, trying to help mitochondria. Right? actually, there are some things about alpha-synuclein that are very convenient for this. And one thing is its shape. This is a kind of atomic level structure of alpha-synuclein.